Welcome everyone to this month's version of the ReproNim webinar. We are so delighted to have a dream team of investigators from the PsychoPy community here to present today. I'll let them individually introduce themselves over the course of the presentation, but I just have to say how excited we are to hear from this group. As we all know, physiological features play a critical role in our use of any technology to assess the brain. It has influences on perception, cognition, emotion, motivation, etc. Even if ReproNim succeeds at getting reproducibility on the data analysis side down to perfect, still variability and variances and things going on physiologically in the subjects that we're looking at are critical. And so without further ado, we're going to have a lovely overall presentation of Physiopy and its relationship to reproducible studies. And I'll hand it over to Stefano.
Thank you very much, Dave. And thank you very much for having us here. I am very happy to be joined by a couple of people from the community. And today we're going to just show you a couple of things that we're doing, that we're cooking up in this moment, and then a few things that are coming up in the future. So I'm just going to very quickly introduce the community, how it started, and why we started doing this. And then Ines and Becca will talk about the community guidelines, basically something that started as best practices, and then we realized that maybe they were not the best, but for sure they were our community practices. And so we rebranded a little. Mary will talk about bitsification of data with VistaBits, one of our toolboxes, and then the one or two bits extension proposals that we have in our hands in this moment. And then Maria will talk about PicTet and Fist to the Noise, which are our toolboxes to process, then use physiological data in neuroimaging. And finally, we'll bring it back here and talk about what's cooking up for the next coming year. So very quickly, why we founded that community and why we keep having a community. Why Physiopy? When we started, which was 2019, physiological measuring and physiological use and physiological data acquisition in neuroimaging was still fairly a niche topic. Experts in this topic were using it and carrying it out, but not everybody was adopting it. And so we started raise a community to raise awareness and spread the use of these physiological data. To do that, we wanted to share this physiological data, share toolboxes that could be easily used to process this data, and then have some documentation that could be used to properly adopt physiological data in a neuroimaging setting. And we wanted to follow the concept of open science to improve the exposition of this topic. And we started at a certain point saying, okay, that's all good and all right, but how do we really work on these topics? Well, maybe we need to get some practices going on. So we started having community practices meetings to reach community consensus about certain guidelines on how to deal with this data from acquisition to analysis. More on that later. And finally, we decided from the beginning that instead of paying people that tell older people what to do, we wanted from the beginning to be a group of people that gathered and choose together what to do instead of the other way around. And so we started adopting a community-driven, bits-based open development approach, and especially aimed at bringing governance back to the users of physiological data and physiological toolboxes. So there's 4 core components in Physiopy. One is the set of easily adoptable toolboxes. Mary and Maria are going to talk about them later. A clear and approachable documentation, community practices based on consensus, and then Inish and Becca will talk about that. And then finally, and most importantly, the community of users, developers, and researchers interested in physiology. All of these 4 core components work together and inform each other on the process. And so, for instance, the community talks and decide community practices that inform the documentation that we write, that inform the way we develop our toolboxes, and vice versa. The community supports the toolboxes that support the documentation and help us increase in the documentation at hand. Using this documentation also informs us back on how to run our community practices and finally get back to users in that way. Now, a quick overview of all the projects that we have going on in Physiopy, looking at the workflow of physiological data acquisition and analysis. So we start from raw data. We don't really cover data acquisition at the moment. But the first thing that we cover is bitsification. And so we have a toolbox that just takes data that is normally in proprietary formats, files, and then brings it back to TSV, which is basically tables that are easily usable by everybody. We then deal with physiological data preprocessing and peak detection. And after that, we deal with physiological denoising. So we create the models to work in neuroimaging with physiology. We're not covering physiological imaging yet. So for instance, we're not covering CVR computations or other types of usage of CVR, of physiological imaging. But we will get there eventually. And around all of these processes, we want to pay attention to quality assessment and quality control. And finally, as I said before, our documentation also describes and guides all of the process that I've talked about so far. And so we have a different toolbox for each of these steps. We decided to be very modular so that people could just use one if they wanted, or all of them at the same time, and interface with other toolboxes otherwise. And so our bitsification happens with Fist2Bits, our peak detection and data processing with PeakDet, and finally the physiological denoising happens with Fist2Denoise. PhysioQC is a new library that we're creating at the moment, and it's still quite at the beginning, but it's what is meant to cover quality control and quality assessment. And finally, our physiological community practices guide the documentation and acquisition of data. We're also working on a coordinated testing suite with other physiological software developers to basically try to get more standardization between all of the toolboxes on what we expect physiological data to look like after we process them. And as I was telling you before, at a certain point, we realized we needed some standardization, especially if we wanted to implement some automatization in our toolboxes. And so we worked on a bits or extension proposal or two. Of note, at a certain point, we realized that we were making things so much complicated on the side of physiological processing that we decided to open a new toolbox for that. Rebranded it in Prep4Phys. Currently, it's the same as PeakDet, the one that we had before, and that was actually very generously donated to us by Ross Marcello at a certain point. But we aimed at bringing PeakDet back to what it was at the beginning, and then continue the development following the newest and latest decisions that we made in the community with Prep4Phys. Now, a couple of information about Physiopy. First of all, I need to give this shout out very, very huge. We were highly inspired by the Tadana community and the Turing Way in what we did so far. I was a member of the Tadana community before founding this community, and the experience there basically informed the first few years of Physiopy. Physiopy was born in 2019. It was a meeting between 9 people in 3 different labs during the DC Code Convergence in 2019. So some of us were in a room there and some of us were on Zoom. But from 2019 to 5 years later, we now count 35 contributors that come from over 25 institutions. Not all the contributors are currently active. Some of them left the community, some of them are on hold, some of them appear from time to time, but all of them made a contribution and a huge step forward for the community. And we normally recognize all the contributions through all contributor schemas. You can see an example here on the screen, and authorship when we have an outcome. And we normally try to acknowledge everybody alphabetically by family name. So all volunteers are volunteering so far. We didn't have anybody hired to develop or maintain these toolboxes. And I have to say development spikes, especially during BrainHack moments. So BrainHack Global or at the UHPM BrainHack. The project had virtually no funds so far, except for a very minimal contribution that a few of us, having done Google Summer of Code mentorship, decided to give to the community. And we are now rediscussing governance. After 5 years, we decided it was time, and we're basing it on the minimal viable governance model. We're also restructuring the contributors guideline, mainly because the original one was not that user-friendly, and we're trying to make it more user-friendly and more newcomer-friendly as well. In Physiopy, we tend to automatize as much as possible. And that's not just because we don't want to deal with headaches on releasing new versions or publishing a documentation or whatnot, but it's also because we believe in the fact that automatization can help lower barriers to contributions, especially if you want newer members that are not experts in coding or not experts in physiology to join us. And so, for instance, we style our code and we automate this stylization with pre-commit so that people don't have to think about it when they write the code, but will appear later. We release on PyPy using semantic versioning, automated by Auto. That is a very friendly toolbox that streamlines releasing with labeling. We assign DOI to every release using Zenodo, or at least we try when it works, and then we release documentation on Read Docs using restructured text and Markdown to make things easy. And finally, all of our software is released under Apache 2.0 licenses, although the practices and documentation adopt ACC BY-SA 4.0. And finally, sorry, forgot about that. Installation of our toolboxes is minimalistic and relies on extra features. That's a decision that we made at the very beginning, in which we decided that we wanted to support people that, if by any chance they had only a watch to run these toolboxes on, they should have been able to do that. And when that happens, normally not all of the features are there. And people that are more feature hungry or wants the cutting edge technology for developing, like for dealing with physiological data, can install those extra feature fairly easily. Now I'm going to leave the next part of this presentation to Ines and Becca, and I'll take it back later.
Hello, everyone. Can you hear me? Okay, so I'll share my screen. Let me know if anything is not working well. So as Steph was saying, I'll talk to you about our community practices together with Becca. I'll show you first the why. So physiological signal integration in neuroimaging studies is great. We can all agree on that, but it brings added complexity because people are usually experts in the neuroimaging part, sometimes not so much the physiological parts. It requires oftentimes extra equipment that's usually it's not even integrated in the neuroimaging part itself. So for example, if you are acquiring in a scanner room, you have to bring equipment that is usually not there probably. And sometimes there are no standard protocols. So people are doing the physiological signal integration in the late years, but it's not an established thing. So there's still a lot to do. And so as a community, we think that joint action is actually the key to make this better and to make this more systematic and hopefully improve the research for all of us. And so these community practices meetings started. So basically, these are meetings that we had and we currently still, it's an ongoing thing. So we are still doing these meetings. And they represent community efforts to try to set this foundation regarding the setup, analysis, and other topics that are considered of relevance. And the purpose is mainly to try to turn each lab's tips and tricks into systematic practices. So there may be a problem that someone has in a lab that some trick from a different lab could easily solve, and we don't know because we're not sharing information. So putting everyone together, discussing these things, discussing the best, or at least the way we are doing it, even if it's not the best yet, can hopefully lead us to a better place. And so the goal is to develop protocols for data collection, synchronization, and analysis, and these turn into the community guidelines. So these guidelines were actually put together in the new version of our website. As Stefano was saying, we tried to make, there was already a a website with some guidelines, but recently we tried to make it more user-friendly and also to add a lot of new information that we gathered along these community practices meetings. And now I'll show you how we did it, and Becca then will walk you through the website itself. So basically, we organized these community practices roundtables attended by researchers from different career levels, from experts to beginners, talking about different physiological related topics. Then we compiled the information that was the outcome of these meetings, and we further enriched it with literature review. And basically, we turned all of that into quite simple markdown documents that were then stored on GitHub and that went live in this Read the Docs format website. So we could have these version control documents that was live and that would minimize barriers for people who wanted to contribute, but still having the information in a systematic way. And so getting into a bit more of detail, just to show you some examples of the topics that we discussed. For example, they're setting up a physiological monitoring system, quality control metrics for physiological data, denoising fMRI with respiratory data, and physiological data is complementary data for functional time series, respiratory signals. These topics came mostly from from the expertise that we had in the community. We decided them through forums and some discussions, but we're open to new types of topics. And actually, the next one that we welcome you to join the community meeting to discuss it will be physiological signal repair, what to do when your signal is bad. And so regarding the contributions, we had contributions from more than 15 researchers from 12 institutions. We are actually currently also establishing the contributors guide for community practices guidelines. And as I was saying, the focus was so far mostly on cardiac and respiratory activity, but we're actually seeking expertise beyond these areas, and people working on different physiological data are, of course, welcome to join and encouraged to do so, because we would like to expand our topics. And now Becca will show you the overview of the websites. So I'll stop sharing and she will share
You can
from.
leave this slide.
Yeah.
Oh,
sorry, Becca. As you
That's
prefer.
Okay. So here you can just see a brief overview of the content that we cover in our community practices, which I'll go over in more detail when I show our website. But as you can see, we go over everything from collecting your physiological data to then using it in your fMRI data modeling. So here you can see our Read the Docs site with all of our community practices. So on the first page, we have just some information about the people who are in Physiopy and what type of data that we work with. So we mostly work with cardiac and respiratory data. And then we also have a section where we just talk about the importance of acquiring data. And we basically go over three main points. So we talk about why or how you can use your data to try to model the effects of physiological fluctuations to denoise your fMRI data. And then you can also use your physiological data as a signal of interest for measuring things like cerebrovascular reactivity with your fMRI data. And then lastly, if you acquire physiological data, you get to monitor the state of your participant in real time, which can be really valuable. And next, we just have some advice for setting up a physiological monitoring system. So there's some information about what equipment you might need, and then also how you can sync that data with your neuroimaging acquisition. And then we have a page on data collection. So we talk about collecting PPG data. And one thing during our meetings that people realized was really important, for example, is mitigating the effects of skin temperature on the PPG waveform. So we have some advice such as providing participants with a blanket during the scan. And then we also talk about tips for collecting ECG data and respiration data. So one other thing that we talked about was looking out for saturation of your respiration belt during your experiments. So making sure that the respiratory belt isn't too loose or too tight so you can accurately acquire your data. And then lastly, we have a section on data processing. So we talked just a little bit about different models that you can use. So both model-based approaches, like for example, with your respiration belt data, you can calculate respiration volume per time or RVT, and then include that in your fMRI modeling. And then we also talk about some data-driven approaches like ICA. So just to go back to this presentation and give a quick summary of the guidelines that we review, we offer a systematic approach for preparing physiological monitoring devices and synchronizing them with your fMRI data. And we focus mainly on cardiac pulsation data and give tips like being consistent with your sensor placement and mitigating motion and the effects of temperature. And then we also talk about respiratory data and give tips like assessing the subject's limits and checking for saturation. And then lastly, we go over data analysis and acknowledge that the methods depend a lot on the research question and the features of the data that you have.
I'm going to jump in. The next part of our presentation is going to include 2 demos, actually. But I hope you'll bear with me for just a moment while I introduce the concept of physiological workflows. So beyond the practices that you just have seen presented, Physiopy, as Steph mentioned in the introduction, is really trying to introduce reproducibility through 2 different ways. The first is through making data standards to store and share your data sets more accessible. And I'll be demonstrating one of our toolboxes that it is designed for that purpose, Phys2Bids, in just a moment. And then we're going to be talking about some tools that actually address how physiological workflows are implemented. There's a strong element of subjectivity and kind of addressing the needs of your data in different ways when you're working with physiological data. And so what we want to do is design flexible tools that make some of these subjective decisions transparent and their implementation reproducible. And so Meriev will actually be showing a demonstration of some of those toolboxes after I finish this part of the presentation. So I'm going to start by talking about the brain imaging data structure, or BIDS format, that Physiopy really truly appreciates, and I think a number of us in this field appreciate, as setting the tone for what a data structure can look like and how much it can contribute to automation and reproducibility. For those of you not familiar with BIDS, the idea is that, especially in an MRI context, there is a lot of variability in what the raw data we acquire can look like. Depending on your study, depending on the sequences and the scanners you're using, depending on your lab's individual practices, you can wind up with a data set that looks something like the one on the left that's really probably only parsable by the people who acquired that data. And so the premise of BIDS is to provide a set of specifications and tools that let you take that data on the left and transform it into data on the right. That's easy to look at, understand, interpret, and share. And so there's a very specific set of organizational principles that govern this transformation into BIDS format. And typically, physiological data has been treated in that BIDS ecosystem as something that is complementary to MRI data. It might be acquired during an fMRI acquisition. It might be acquired during another MRI sequence. But usually, if you're looking for data that someone has acquired during an fMRI sequence, for example, and it's in BIDS format, you'll find it in a functional folder, and it will be something like cardiac or respiratory data. So there's a part of the BIDS specification that governs how to store these types of data. And Physiopy has developed its own toolbox, Phys2Bids, to help people convert their raw physiological files into this greater BIDS format. We support a number of the most commonly used formats for physiological data. There's different third-party data hardware and software acquisition systems, namely BioPack and AD instruments that we support. We also support some physiological data recorded directly by the scanner. One area that we're looking to expand into is Siemens data recordings as well. So I'm going to switch and actually show you just a little bit about what Phys2Bids looks like. Can you all see that I've switched to the command line? Hopefully, perfect. Okay, so I won't show you how to install Phys2Bids because we're trying to cover a lot today, but it's very simple to do. You can do a pip install on your own. All of the material for this demo is also going to be available in a repository that we'll share with you. I think we'll share that in the chat, but it's also linked in the slides. Oops, excuse me. So just to show you briefly an overview of Phys2Bids running at the command line, if I just run a help argument on Phys2Bids, you can see it's very well documented how to use it. We also have, though, a read the docs specifically for this toolbox with much longer tutorial than the one I'm going to give you here. So the first thing we can do with Phys2Bids, you can see I have in this folder just two files right now. One is a Python file, and one is this .acq file. That's a physiological file in a proprietary format. So I want to use Phys2Bids to give me just an idea of what's inside that file. And I'm going to use the info flag so that, again, Phys2Bids knows right now I'm just interested in taking a look at what's in the file. I'm not actually going to start that process of converting to bits yet. And you can see at the command line, it's going to tell me exactly what this tool is doing. And importantly, it's going to take a look at the file and it's going to tell me some of the different types of physiological data it's finding in there and their sample frequencies. And it's reading that information directly from how things were labeled in the original file format. It also creates this nice plot so that we can get a sense of what that data looks like. You can see here there are 6 different channels of time series within that data. The first one is some respiration data. The second one, the channel name is labeled pulse, but this looks also a lot like respiration data. And here there was actually an error in how the file was labeled at the time of recording. So that's more respiration data. The third channel, you see a number of very sharp pulses, and that is data recording the time of each MRI trigger that's being received alongside the physiological data. This is something we really strongly recommend. Acquiring this data lets you synchronize very precisely to your fMRI data. The last channel, channel 4, is PPG, or a kind of pulse or cardiac data, and the last 2 channels don't seem to have been used for this recording. So now that I've used Phys2Bids to give me an idea of what's exactly in my data, I've taken a look at my data, I'm going to go ahead and use Phys2Bids to actually take this data and convert it into bits format. So I'm starting out similarly. I specify my input file, and I'm going to specify a bids folder to output. It needs a little bit of information about the accompanying MRI sequence in order to set things up in a way that are going to adhere to whatever functional data it's accompanying. So in this case, it wants to know the number of time points in the accompanying fMRI scan. For this case, it's 60. The TR of that MRI scan, in this case, it's 1.5. I'm specifying that I only want to convert the first 4 channels of this file because those are the meaningful ones. And I'm also going to rename those to make them a little bit more coherent to people who might use the data after me. So I'm going to name them respiration 1, respiration 2, trigger, and PPG. Lastly, I just provide a little bit of information about the bids directory structure that I'd like, including the subject number, the session number, and some task information that I've customized in the accompanying heuristic file. And we have a full tutorial on our documentation for the toolbox that will show you how to set up a heuristic file. But basically, it's just going to take your file name, and from there, you can create a set of rules that will tell it which other bids fields to populate in any given way. So I'll go ahead and run that. It won't take very long. One of the important things it's doing now, beyond just looking and seeing what's in my file, is it's going to count the number of pulses that it detects in the trigger channel. And if it detects a number that's different from the number I originally gave it, it's going to raise a red flag and let me know that. So this is a really important point for reproducibility, where you're going to be able to check that the physiological data you're bidsifying is going to match the functional data in the way it should. So here it tells me time points expected 60 and time points found 60. That's great. We also have different capability, like being able to split longer physiological recordings acquired during an entire MRI session into specific sequences. So if I go into the folder that was generated this time, I have the same folder as before, where it will show me just what my data looks like. It's also going to take a closer look at the trigger channel and show me where it found triggers starting. So again, this is a point where you can check that your code is doing exactly what you want it to and that your physiological file looks the way it should. And then other than this, the data is now in BIDS format. So you can see I have a subject folder, session folder, and functional folder. And here, if I go in to the files where my data were actually stored, you can see that I have metadata that's been generated to accompany each file. So this metadata is telling me that in the file with the same name, the sampling frequency is 625 hertz, the time it starts at relative to my MRI file, and the types of data that contains. You can see it's the same for the other file that was generated. So this is just a very brief snapshot into what Phys2Bids can do, how it's been set up to take that proprietary data and turn it into a format that's easy for other people to access and understand, and importantly, for software to access and understand. Oops, excuse me. So this is just the start of a physiological data workflow, this conversion into BIDS format. And it's a really good place to start as far as being able to create open software that can create the rest of your workflow. I want to briefly touch on a few points that are involved in the rest of a physiological data workflow. And I want to emphasize that each of these following steps is another source of variability, is another way people can implement things differently or make different processing decisions. So to start with, when you have your raw data, you typically need to do some filtering and artifact handling to actually make your data intelligible, and especially in an MRI environment to deal with any artifacts you may also be acquiring with your recording. The second step might be to perform some kind of feature extraction and validation on that pre-processed data. And for this signal, it's a PPG signal. So what I'm really interested in is peak detection. I want to identify peaks that correspond to heartbeats. The last thing I might do with this data, once I've taken a look at these peaks and I've made sure they're where I want them, is to calculate some kind of metric. In this case, I've calculated heart rate variability. And the goal here is to use that metric in a way that it provides some insight into my fMRI data. So in using that data, I want to be able to potentially take it, share it, make it intelligible to other people. So the underlying question in all of these physiological processing steps is, how do we share the workflows and make those outputs something that is useful to other people? And again, Physiopy is approaching these questions two different ways. We want to create open software tools so that people can perform some of those operations like peak detection in a very transparent way. But we also want to create data standards that let you share these workflows and their outputs at different stages. So we can think of this as facilitating reproducibility checks across different sets of software and across different types of analyses once you have those final metrics. So returning to the idea of BIDS and standards for data, as Steph mentioned in our introduction, Physiopy and a number of collaborators who are adjacent to Physiopy are helping to write some extensions to BIDS data standards that will let us increase reproducibility for physiological data. We've boiled them down to some key points, one of which is that we're interested in how this data structure can apply to physiological data even if you're not centering MRI data in your analyses. We're also interested in what metadata needs to be included alongside physiological data to increase the interpretability and usage of that data. So something like if you're acquiring PPG data from an earlobe, a finger, or a toe, that will significantly affect how you incorporate it into your later analyses. We also want to create better tools for showing the provenance of some different derivatives, some guidance for outputting different types of derivatives so that it's very easy to share, for example, a set of peaks that you've identified in your PPG data and allow people to calculate different metrics. So we're currently splitting these into 2 proposals for bids. If you're interested in collaborating on those, we'd be really happy to have you at our working group meetings. And with that, I'm going to pass things over to Maria for our last demo.
Thank you. So I'm going to continue from where Mary left in the code demo. So this notebook is, the link of it is accessible through our physiopy.github.io website. So you can just click here and see that it leads you to a repository where you can find the Jupyter notebook if you want to rerun it during your spare time. So I'm going to start with importing the libraries that we're going to use. And we're going to continue from the data conversion step and tackle step 1 and step 2 with PigDet. So first, we're going to load the data that we have converted into the BIDS format using the load from BIDS function from our Physiutil library, which is a library that is basically a common tools for the Physiopy suite. And we're going to see what's inside of our data. So we can see that it is a dictionary containing multiple elements, and each element is one of our physiological channel. And for the rest of the tutorial, we're only going to focus on the PPG data. So we're going to save that data into our variable here. And we see that we have a physio object. The physio object is what PigDet use mostly to save the data and different attributes. So for example, the data that we can access, calling the data attribute, and the sampling frequency that we can access, calling the FS attribute. So now that we have our data, we're going to visualize it using the plot physio function in PigDet. So we see our time series here. We see that we might have some drift in our signals, maybe some noise, some motion. And so after that, we're going to try to clean our signal to try to remove those artifacts. But first, we're going to start downsampling our signal because it was initially sampled at a frequency of 10,000 hertz. And now we're going to downsample it to 250 hertz. And one of the pretty cool feature of PigDet is the history attribute that can save all of our steps that we're applying on our physio object and the parameters that we're specifying in that step. So as Mary mentioned before, when we are pre-processing our physiological data, there's a lot of degrees of freedom that researchers can have to specify, for example, to what frequency they want to downsample or resample their data, what kind of filtering they want to apply. So there's a lot of variability there, and the history is a way to track those variabilities and save it to be able to reproduce it afterwards. So now we can apply our filter. We're just going to use a bandpass filter using the filter physio function. And we see here that in our history, we have added the filter physio step with all the specified parameters. And now we're going to check our signal. So here I'm just like looking at the first 30 seconds, and we see where we add like that little drift. Now it has been corrected. So we are happy with the current state of our signal, so we can proceed to finding our peaks. So finding our peaks is an important step that will allow us to eventually compute other metrics such as heart rate and heart rate variability. So we're going to do that using the peak find function, physio function. And here again, we see that the step has been added to the history, and we can check our peaks again. So all of that is really cool because we can just automate that. But the thing is that sometimes we might, our algorithm for peak detection might misidentify or unidentify some of our peaks. So manual check is really crucial in that case. So there's a really cool function in PeakDet that allows us to manually correct our signal. So it's the edit function, edit physio function. So basically, we're going to be able to interact with our data in that way. And here we see that we still have that that motion here, that the little noise in our signal time series at the end of our time series. So we might just want to remove those peaks. So how we're going to do it is really simple. We're just going to do a left click, select the window, and then the peaks have been removed. And if we want to, for example, add some peaks because our algorithm missed the peak, we can just use our right click and it's going to add the peak. So once we're happy with that, we can close the figure using Control Q. And now if we check our history, we see that all of the peaks that we have manually removed, all the indices of those peaks have been listed in our history, as well as the peaks that we have added. So now that we have completed the filtering step and then the feature extraction step, we're going to save those data using the save physio and the save history. So we're able to save the physio object in a .phys format and the history in a JSON format. So now that we have done that, we're at the third step, so metric calculation. For that, we're going to use phys to denoise. So we're going to import what we need. And then we can load the physio object. And what we see is that when we load the .phys file, we also load the history. So we can really keep that throughout our workflow. And we are ready to compute some metrics. So we're going to just compute here some the heart rate and then the heart rate variability. It will take a little bit of time. And what we're going to be able to see once that's going to be completed is that the output of those functions are the physio object. Again, that is tracking what we have applied, what function we have applied to those objects. So here, for example, we see that we have the computed metrics attribute that where we can see that we have computed the heart rate on it. And then we can access our data here. So basically, it's a NumPy array containing the heart rate and the convolve heart rate with a cardiac response function. So we can see what's the output. And then we see in blue the arch rate, and in orange the arch rate variability. And now we are able to save our artwork. And we're going to be able to use those inputs. For example, we're going to be able to load them and include them in our GLM, for example, to regress out that from our fMRI data. So we see now in our directory for our derivatives, we have our preprocessed signals and then our metrics. So the arch rate and the arch rate variability, the raw one, and then the convolve version of it. So as Stefano mentioned, we are also working on different libraries and like improving those libraries. So if you want to contribute, please check out our contributor guide. And just want to say a big thank you to all of the people that have contributed to at least one of those libraries.
Some of which are actually at this meeting. So thank you very much once again. If I have 5 minutes more, I'm going to be very quick because I know we're going a little bit over time, but I wanted to just show you what is up next with our community. So, you know, if you get inspired and you want to contribute, we would appreciate that. So first of all, as we were saying before, quality assessment and control is an important part, not only of reproducibility, but also in saving time and effort in acquiring good data and sharing good data. And so we put together a library that is called PhysioQC, that is heavily inspired by MRIQC and is actually using the same underlying technology. And to give some reports about the quality of the data, for instance, average peaks, formats, for instance, some TSNRs and other metrics that can be used to understand if the data is coherent. We're also working a little bit our GUIs to make our software more accessible and give it more features. And so, for instance, this is the future of prep for phys, in which we want to just give as much as possible informations and features to users to look at the data and work with the data. And working with the data manually was something that we realized was very important during our meetings as much as it was to look at the data together. And so, for instance, that's why we moved from big data that looks at one data at a time to more complex types of data formatting. We also have more community practices, as Ines was saying. We're going to talk about response functions and convolutions, physiology and vigilance, physiological for confound, especially in resting state. We want to talk about other modalities that are not just the ones that we know about, but for instance, starting with ECG, PPG, and heart rate variability, blood pressure and cerebral autoregulation, and brain-body interaction at large. And more, if there's somebody that is interested in talking about more. We have also some announcements coming up. First of all, we want to improve our guidelines, as we were saying before, with other modalities, starting with eye tracking and ADA. We currently lack the information for that, though, and so we want more contributors for it to suggest tips and tricks on how to collect these modalities, especially with neuroimaging data that is not just MRI, but also either on its own or other types of neuroimaging like EEG. And adding guidelines and support for this type of data means also moving toward integrations with other softwares. For instance, there's a software that is handling very well automated or semi-automated physiological editing, that's NeuroKit, and we were gonna move toward integrations of our software with them. We also want to be integrated or prepare the integration for NIPREP, SAFNI, FSL, and other types of neuroimaging data processing softwares. We're going to extend our support very soon to modalities that are not there yet. And so, for instance, as Mary was saying, Siemens is something that we're seeking, like data from Siemens is something that we're seeking out to be able to implement it into Phys2Bits. Betalino is another type of acquisition format that we're not covering, but we want to start covering. And finally, non-MRI modalities wants to be supported as well. And that might happen as soon as tomorrow, but otherwise after our BIDS extension proposal gets through. We also want to give more functionalities to our users. Semi-automation is one of them. We're talking about semi-automation because, again, we think that looking at the data and do peak detection, supervised peak detection is important. We want to make our data more BIDS applications-like, and finally integrate all of our workflows in one for the people that are interested in moving from BIDSified data to the end result in one stroke. One shameless plug here: more voices and more diverse expertise improve our community. And as I was telling at the beginning, we're really not here to tell you what to do. We want to figure that out with everybody and find consensus in what should be physiological data imaging and preprocessing. So please join us. It's very important for us to understand also other types of informations and types of expertise that are not just MRI and they're not just respiratory or cardiac. And please join us if you're not a physiology or a Python guru. It's okay. We need you. We need people that are experts as much as we need people that are not experts to ask the good questions and to help us develop good software. If you want to join us, please send an email to physiopy.community@gmail.com. And please join our newcomer meetings that is coming up in a couple of weeks from now on November 14th at 4 UTC. It's going to be a Zoom. In order to avoid bombing, we're going to provide links by email. And if somebody else can please send, we prepared a quick form to fill in the chat. I see that that's happening. Thank you very much. Or send us an email through physiopy.community@gmail.com and that would work. And for my moment, that is all. Thank you very much to all of the Physiopy contributors that in these last 5 years made this community what it is today. And to the ReproNim crew for having us here and be able to talk about our physiological community. And finally, thanks to everybody that is looking at this video or has been listening to us for the last hour for your attention. And if you have any questions, opinions, objections, or you want to have a quick discussion, we are happy to start it.
Great. Thank you so much to all of you for that lovely presentation. Again, just a lovely tour de force on all things Physiopy, which are very important to us as a whole. There's certainly other questions around I'll start anyway. One of the experiments we do is with 6-month-old sleeping babies, which are very hard to keep asleep and very challenging auditory experiment. And putting respiratory belts or even keeping a pulse ox thing on a finger is just one step beyond what my poor clinicians can do, keeping these sleeping babies operational from the functional MRI point of view. Is there any work that you can foreshadow on sort of remote sensing of some of the key physiological types of things? Can you get respiratory from a video feed of other features without, sort of more active, belt-like things? Is there, even through closed eyes, eye tracking types of things that can be done from video feeds? Anyway, just a question on sort of remote sensing of some of the physiological features.
If anybody else wants to jump in or answer into this question, I'm very happy to hear what everybody else has to say about it. To my knowledge, there are a couple of things that are developed now. I am sure that Siemens, for instance, has a way to record respiration that is mainly visual, or sorry, not respiration, cardiac pulse that is mainly through feedback cameras. But there's also some other libraries that we don't cover or we're not integrated with yet that use deep learning to do that. So that could be a possibilities. But I wonder if Becca, Marie-Eve, or Mary have more experience with that.
Yeah, I think you were alluding to a study that was learning respiration data from fMRI data using deep learning. So that's definitely one potential option. And I've been working with trying to collect data in stroke participants and kind of had a similar experience that you were talking about, where just due to the limitations of the study and the fact that there's already so much going on, I can't collect any physiological data. So I've been trying to explore other ways to look at physiological signals in the data. So for example, I have the participants do a breathing task at the end of the scan just to try to cause larger physiological fluctuations in the data. so that it's easier to model them. And then there are also like some tools that you can use that don't require physiological data to be inputted, but can still look at physiological effects in the data.
Great, thanks. Other questions from the audience?
We have a question in the chat as well. Looking at it's from Christine. As you work on emerging community standards, are we looking to EEG community examples like the Artemis project, as well as BIDS, of course? And I did mention I'm not familiar with the Artemis project, so thank you. We're looking to support more physiological data integration in electrophysiology. My lab does EEG, FNIRS, as well as fMRI, for example. So both of those things are relevant to us. But really, we just need more people who are experts on EEG to come join Physiopy.
I have to say,
answer-
Ines is our EEG expert in-house, but I'm sure she would be happy to start talking with somebody about how to deal with stuff, as most of us are mainly MRI based.
Yeah, I work with EGF MRI and still linked to the previous question. For example, when you acquire EEG, at least in adults, I don't know how it would go for babies, but we actually use an ECG and we have been deriving also respiration from that ECG. So there's another way, if you have the ECG, to actually retrieve respiration. But yeah, I would love to have more people working on EEG to join. So
Great.
you're all
That's
welcome.
That's exactly why I asked, because there's so much overlap, right, with the ECG channel, and yeah.
Okay, interactions forming, I see it happening. Felix has a question.
Yeah, and thanks for the presentations. They're very informative, and the project is very impressive, what you created and how it looks like now. My question is, let's say, a little naive, and from my historic, I never used the database. I didn't acquired it myself like 10 years ago, and then it was basically trying to use the biggest data set that are there. And I found sometimes some small attached images that I knew were physiological data, but the problem I had that you seem to solve, I think you solved it mostly, is how to use it, how to transform it, make it usable, and then use it in the analysis. So my question here is then, can you recommend, so now is more how can I start? I mean, which are the bigger data set where there is data, and it would be basically a good starting point for one bigger data set? Because I know I can remember faintly in HCP and others, it is there. It's still the thing, nobody uses it, but I think that one of the ways of advertising it would be to transform the data, attach it in a data set, and make it easy for the people to use, and of course show it actually makes a huge difference as well. Not only increase N and then you have it, but actually use this and you are better, right? And not only for your study with like 20, 30, 50 people, but as well for the bigger data set. So what are your recommendations that I can start following basically, or actually using your tools?
I can tell you there's a data set that is coming up that is going to contain a lot of physiological data measurement in MRI. I see Basile and Marieva here, and so they can tell a bit more about that. Pre-existing things, unfortunately, a lot of data that is acquired is either, as you were saying, non-transparent, or the quality of the data was not necessarily checked because it was not necessarily the most important part. And so HCP has some data. I believe Study Forest should have also some data like that, no? Or, I mean, Felix, you might know better than me that, but I believe Study Forest had some physiological data on that. And otherwise, other big project like MyConnectome has physiological data. Some other recordings or smaller data set on OpenNeuro feature some physiological data as well. I'm thinking Excalibur in this moment is one of them. It mainly concentrates on physiological data, and it's being released a little bit by little. But yeah, that's what I can think about.
Yeah, great. And
You can also search on some platforms like OpenNeuro. Like you can add a keyword like PPG, for example, and people are usually very good about labeling their data.
Now, to find data, that's not most of the smallest problem. But then to I would like, so how it seems very easy that I can ingest it in the workflow and use it for that. My specific question is quality control actually for structural imaging data. Now, we don't have movement data, but that could actually solve the problem because we can infer movement from use the basically noise channel there or something else and have direct measurement or some numbers to use in addition to, let's say, MRIQC and other measures we have to associate these images, in addition to look at it in the rest. So I'm in a project and starting this, and I think that's very promising. But I think I'll ask you later then as well in chat because I think there's huge promise as well automatizing it for huge data sets. So very inspiring. Thank you for all this.
Thank you for the questions. I know we're a touch over our time. Any burning questions before we go on? Hearing none, again, everyone is quite accessible in the interwebs. So any questions, this conversation, of course, can continue. So I'd like to thank you again for the presentation. We'll see you all around the reproducible community and the open science communities at all of the hackathons and workshops and things like that. So looking forward to continuing the conversation.